the diagnostics for it became more, so we're seeing more of it globally. The WHO put out a report in the last few years. They are saying, many more STECs or EHECs are the problem, and these are the things we should focus on. And it doesn't matter if they're 0157 or one of these other serogroups. Anything that's making Shiga toxin and can bind inside of a person and cause an infection is a high priority. And so that shifting focus and definition, it kind of keeps broadening the net of STEC that we want to try and monitor and keep out of beef and other foods.
Welcome to the Beef Podcast. I'm Travis Mullenix, and today I'm excited to have Mick Bosilevac with me today. Welcome to the show, Mick.
Glad to be here.
So a little different topic for me, a little something out of my wheelhouse, and talking about food safety and pathogens, etc. And so excited to get discussing some of this. That's totally different than stuff that I think about and that I do on a daily basis. So Meg, just to give us a little background, I know you work at Clay Center at the ARS station there, Meat Animal Research Center there in Clay Center, Nebraska. Just give the audience the background of what you do and how you got there.
All right. Well, I'm a research microbiologist in the meat quality and safety research unit there in Clay Center. Work for ARS, the Ag Research Service, part of USDA. Our research unit is focused on quality and then safety. Safety is my side of things. I was kind of recruited back into the unit 25 years ago when more molecular biology skills and tools were needed. So back then, there were a lot of food microbiologists, but not many people applying, things we take for granted like PCR and these things to detect pathogens. So I was kind of hired in to bring in that sort of set of tools and apply them to food safety. Before that, I had not much of a background for food safety. I came from clinical and diagnostic lab background and had actually got my PhD in cancer biology. So, had the tools, but then was recruited in and kind of taught food safety and meat processing and said, no, there's the problem, help us fix it. And so we've been working on that ever since, focused on 015787, which was and still is the biggest problem on the E. coli side of things.
That's great to have you, your expertise, and to learn more about food safety. And just to touch base, this is a sponsored podcast by bioMario that's sponsoring today. So jump into some of the discussion points that we want to discuss of, how did you become involved in some of this food safety research? You touched on, getting recruited for ARS and not coming from necessarily this background, but can you touch on how you got really involved in food safety?
This current position or this position, being the research microbiologist, working towards, on food safety, controlling contamination, mostly in beef, a little pork, a little lamb, but, focusing on controlling pathogens, learning from, where animals are raised, all the way through how that influences something that can contaminate food that's heading towards a consumer. So, that's sort of the problem put in front of me, as well as the high priority of 015787. It was, causing outbreaks and recalls and, can be a very severe pathogen. And so that was, large focus, by the agency as well as, the regulatory side of things as well, and the beef industry themselves. So, we have a good working relationship with those different groups, and that kind of, was what got behind all of it.
For over 60 years, bioMario has been a world leader in diagnostics, now revolutionizing the beef and meat industry with augmented diagnostics. Addressing critical challenges such as food safety risks and recalls, production costs, and waste, as well as environmental and consumer concerns, bioMérieux offers a fresh approach that goes beyond test results. Their unique portfolio of microbiology testing, genomics, data solutions, and expert insights help with pathogen detection, enumeration, and root cause analyses. By providing improved risk anticipation and comprehensive data, bioMérieux empowers beef processors to confidently navigate today's complexities.
So what exactly are STEC or STEC, as you told me that you've kind of switched how you call that, but what is STEC, and why would anyone be concerned with?
So the STEC, or STEC, so it's the Shiga toxin producing E. coli. So there's your E and C. So STEC, sometimes pronounced STEC. And I'll actually maybe use the word EHEC here when I talk about some of these, because there's a lot of STEC out there, but EHEC or enterohemorrhagic E. coli, and those are the ones that cause the most severe disease. So 015787, as I mentioned, it is a STEC because it's a Shiga toxin producing E. coli, but it also carries some other virulence factors and things that make it more pathogenic, much more able to cause disease if you were to consume it. And so those and a few other E. coli then we refer to as the EHECs. And so the STEC produced that toxin. Toxin can can get into your bloodstream. It can cause diarrhea, bloody diarrhea, kidney failure, and if untreated or in an elderly or young child, it can be an overwhelming infection and lead to death. And so it's an important pathogen. Fortunately, we don't see a lot of it, but when those infections occur, they get a lot of attention, and just because they can be such a problem.
Is it becoming more of a increased awareness of it, or increased public health concern of some of these challenges?
Yeah, so things started with 0157 back in the 90s with like the first Jack in the Box outbreak and things, and focused was on that, and that's kind of when I started. But then somewhere in the 2000s, there, CDC released a study where they saw that they were starting to see these other non-0157 STEC, and so these E. coli make Shiga toxin. They carry some of the virulence factors. And when CDC was looking at all their attribution data, they said, oh, look, here's 0157, and now here's this other group of 6 serotypes or serogroups of E. coli that are producing just as much disease as 0157. It's not as severe. There's only been a few deaths related to the non-0157s compared to 0157, but they can still cause similar disease. And so that awareness of, looking for 0157 and then finding these additional STECs has kind of brought that, more and more, it's a more common test in clinical laboratories to screen, feces samples from ill people for Shiga toxin. And then if they see the Shiga toxin, figure out why it's there, and so, the diagnostics for it became more, so we're seeing more of it. And then globally, the WHO put out a report in the last few years where they are saying, many more STECs or EHECs are the problem, and these are the things we should focus on. And it doesn't matter if they're 0157 or one of these other serogroups, anything that's making Shiga toxin and can bind inside of a person and cause an infection is a high priority. And so that shifting focus and definition, it kind of keeps broadening the net of STEC that we want to try and monitor and keep out of beef and other foods.
So what's driving these changes?
Part of it is testing and monitoring. And part of it is public awareness or pressures, outbreaks, recalls, these things get people's attention, wanting to know what they are. And then just a lot of sequencing information as, more and more of these isolates are found, there are more genomes are being sequenced, they're more being studied. And so we got this large and growing repository of data that can be examined, and we can start looking at E. coli, the Shiga toxin E. coli, ravage EHEX, and start determining, why are these more pathogenic? How do we focus on them? How can we control them? And it's kind of a growing area, of growing or getting a larger group of things to try and control, or they're being recognized easier, I guess, is one way of saying it.
As a research scientist at Clay Center, how do you share your research results? And I'm assuming these are going to like the more of the slaughter processing type of facilities research, or is it taking a step back and even on the feeding phase?
A little of both, actually. As I'd mentioned, we work with the beef processors, as well as some feeders, looking at ways of trying controlling these E. coli. I guess we should mention, as part of background, they live naturally in cattle, and they don't make cattle sick. So they live in cattle, they live in the feedlots, and from that point on, they can carry them into processing plants. They get transferred to the carcass, then they can stay associated with the meat and end up in finished meat products. And so, our product, when we're working on these things, we've, have worked with some feedlot and feeders, and most of it is more on that processing side, because all the cattle do funnel through, a harvest facility of some sort. And so that's kind of, the pinch point where we can start applying ways to control these E. coli. Since those are sort of our stakeholder groups, feeders, as well as, the processors and regulators, my science and our results, first of all, there's typical scientific publications, but also we do work directly with processors, the big 3 or 4, as you would say. We know all the, people who are the sort of VPs of safety in those companies, have a good working relationship with them. Tell them what we're learning, or they'll come to us with a problem saying, how can we control this? We've found a bunch of E. coli or had an incident, or a situation. Let's learn from it. And so we'll work with them, both ways, doing some studies, and they're really good about, knowing that we're going to be, impartial when we, examine either some way of trying to decontaminate or control E. coli with an intervention, or compare some ways of detecting it. So we give them those results of what we learned sometime, and often that's well before it ever gets into publication. They're getting these information from us straight from the lab many times on what we're finding and how they can start working to keep these things under control.
And assuming it really helps for your end of not only being at Clay Center, but being in a big beef state. That helps really disseminate and being able to get those results out.
Yeah, we've got 3 or 4 small processors available, and each of the big 3 are present within a few hours of the research center. And by having that good relationship, they will actually, we are allowed to get into the processing plant and collect real samples, from carcasses or during hide removal, or and through. So it's a really good way of getting real relevant samples, and not just things that we treated some E. coli on the bench in the laboratory. We killed it, great, but if we take this into a real world situation, what can we learn? And that's probably the main bread and butter, the main impact of what we're doing is we can get access to those real facilities and see, give measures on how these things work.
So what are the challenges that beef production has faced with this?
The biggest one, there's the pre-harvest side of things, trying to understand how 0157 is living in cattle, and if there's any way of reducing it in cattle. There's some, vaccine approaches or sort of probiotic feed additives that have different effects on reducing it, but they still get carried into processing. And so the challenges there then are finding interventions. Organic acids get used a lot to clean carcasses and things to, remove E. coli, injure it so that it can't grow or, stay with the meat as it's further processed.
As was detecting a challenge?
Yeah, and then, so if contamination does occur, then the other side is properly detecting it. And, basically every 2,000 pound combo, either individually or in batches of 5, get tested, all that beef trim before it's ever used for ground beef. So we're talking thousands of tests performed to ensure, safety systems at a processing plant are controlling E. coli, and then to make certain that any subsequent products don't have it. And so, as I mentioned, there's the 0157 and these non-0157s. And 0157 is kind of, a very easy target. It's a 0157H7, so it's a very specific serotype. So we know lots about it because we've been studying it many years. Hundreds of genomes of that organism have been sequenced. So by comparing all of them, some very good, targets within the DNA targets that are very specific and sensitive, let us detect only 0157H7. And then on top of that, they're very easy, they are very easy now to confirm that they're present, right? So I can, have my beef, I screen it for 0157H7, I get a positive signal 90-95% of the time, then I can find it because we know that it has a specific, phenotype on certain augers. They stand out on the plates, and we look for them on Petri dishes. We've got tools to help capture them and give us positive culture results. When you move into those other non-0157s, now you've got 6 serotype groups, and then each serogroup may have, a few serotypes within it. So now, we're talking going from 1 to a group of about 12 or more E. coli that we're trying to capture in a single test. And when you're trying to screen for those sorts of STEC, it gets much more difficult because you've got so many things, so many differences. All you can start doing is looking for the targets that make them a STEC, the Shiga toxin gene. And I mentioned to cause disease, they need to adhere inside your intestines. If you were to swallow one or eat some contaminated beef. So that other gene is typically intimate, so forms an intimate attachment to the inside of the GI tract, where it then causes disease. And so those two targets, Shiga toxin and intimin, are the way that we approach testing or detecting the STECs. And that's the big challenge then is the industry will test everything for 0157 to ensure it's not present, and more importantly, show that their systems of interventions and processes are controlling E. coli. We know that the non-0157 STEC react the same to peroxyacetic acid or lactic acid treated to a beef carcass, and all the different hot water wash, whatever is in place. We know that 0157 and non-0157 E. coli react similarly. So beef processors monitor the 0157H7 to ensure they're controlling all the E. coli that's coming through the system. These other STECs, because we don't have a good way of determining if, by those two genes indicate a real pathogen is present. I guess, the challenge is that, I'm going to start talking about the genetics of E. coli a little bit, but there's the STEC, they produce Shiga toxin, and then there's the EHEC, they have the Shiga toxin and that adherence gene, intimin. But then there's, STEC out there that only have Shiga toxin, and they don't have a way to attach inside a host, so they don't really cause severe disease if they were to pass through you. And then that adherence gene can occur by itself, and another group is E. coli. And so those E. coli don't really cause severe disease as well. But both, those with the Shiga toxin, those with intimin, can be part of the background contamination if you're testing beef. So, just a few E. coli in there can send what looks like give you a positive test for that Shiga toxin and that intimin. And that is why, there's a resistance to, even the regulators have trouble confirming their tests when they start testing for STEC versus 015787. And so, industry is focusing on controlling, monitoring 0157. The regulators are testing for 0157 and the non-0157, and that's the challenge, is to come up with a good way of improving or finding the next steps on increasing the reliability of, Shiga toxin non-0157 testing.
Since it's such a challenge to detect, what's your research been focused on of how do you detect these or increasing the ability to detect these?
Yeah, so what some of the mentioned, we have these large databases of genomes, and once you start kind of mining through all of that, you can find a few genes that will start segregating with, say, all Shiga toxin, Inum, E. coli. They're not absolute, but we're following up on these various virulence factors that kind of associate with the 157 and the other severe disease-causing STECs. And there's some genes and sort of pathogenicity islands that will follow along more often with things that have Shiga toxin in them and are less likely to be present in something that only has Shiga toxin, or only has intamin. And so that's kind of where we've been working, is looking at various virulence factors. There was a study we did for the beef chuck-off many years ago. We kind of dug through all the literature. We found like 20 virulence factors, and we just started screening carcasses of the processing plant just off right after hide removal, so that these were contaminated carcasses or likely contaminated. And then we took those samples and tested them, and we found that the more virulence factors we found, the more likely we were able to confirm there was pathogenic E. coli. There was no golden ticket there or key, but it was just kind of like, the more you find, we see 10 to 15, we can culture confirm this. If we see less than 12 and 8, we're not going to be able to culture confirm it. And so, we knew there has to be, some value in sorting out which the best virulence factors might work. And so that's kind of, what our current work is looking at, is what virulence factors can help improve detection and, and better ways of, say, enriching or processing the sample to collect, to get to the point where the E. coli get to a good detectable level, and you can sort out whether or not they're pathogenic or just sort of a mixture of E. coli that are there.
Yeah, what have you found in strategies or processes for beef processors to ensure that their products are safe as possible?
Well, yeah, as I mentioned, safest products are keeping E. coli out of the process, out of finished products, and so there is 0157 monitoring. The other part is some of these virulence factors. A commercial test has come available recently. It's called PEC, or pathogenic E. coli, and we've analyzed and did some work on that test, and actually have a paper out in the literature on it. And this has a test for a couple virulence factors that associate with those Shiga toxin M positive E. coli. And if you use that test as your sort of your screen, it can tell you whether or not product is free to release. Because if you don't have the virulence factors present, the PEC test, then product is beef product, ground beef trim, that's free of Shiga toxin M and E. coli, and it's good to go. If it's present, then further testing, some next steps, look for Shiga toxin, look for the intamin, look for which O groups or serogroups of E. coli might be there. So, using the virulence factors as initial screen has potential, and it's actually part of FSIS's official method right now, where they use this PEC test along with the first STEC screen to identify samples they want to further evaluate. And what we found was, by using that approach, The markers are in all EHEC we tested, right? So everything that was Shiga toxin enamine, no matter what serogroup or serotype of E. coli, very common ones and very unusual or rare ones we don't often see, they were all positive with the test. We tested, lots of things that were STEC, Shiga toxin, but lacked enamine, so therefore not very pathogenic. They all lack these targets. And then there's those others that carry the enamine alone, that other second adherence factor. And in that case, we could see maybe half of those E. coli carry these pathogenic factors. And that's partly because they're involved in some of that, or they've evolved along with that adherence factor as part of the disease-causing ability if Shiga toxin is present. So we've got sort of a low level of background. And so when you use the PEC test, you're going to end up kind of cutting down, say, implicated or potential positive things in about half.
So what's the impact of your results, your studies have on STEC testing?
So our impact of our results, some of the early on reports, test and hold programs, testing for 157H7 and not releasing any product until it's confirmed negative. We did some of those very early tests 20 years ago, and that really influenced the process and how those were put in place. More recently, we've got, like I mentioned, we've studied the PEC test. We're looking at other tests and methods of approaching testing that can tell us whether or not Shiga toxin and enamine are in the same E. coli to have a more confidence on what we're testing. Whether or not the positive result you get is a true positive. And then on the backside of that, we're also trying to come up with better, improved ways to isolate or identify that STEC if it's present, get it back out or find it. And in that case, we're finding there's a bunch more of these potentially EHEC out there that may end up being, part of the regulatory definition someday. But that's kind of, where the impact is going. A lot of the mentioned FSIS is using the PEC, and a lot of the, their initial decision to investigate that test and evaluate it was work that we had done with and for them, part of an interagency agreement and data sharing. They provided samples, we provided analysis and further workup on the samples. So it helped, you got that test, into their hands. They're using it, and then, we've been presenting it to, at say, a beef checkoff or at a beef safety summit meeting to, kind of explain to the industry this is what the test is, because since FSIS is starting to use it, there's a lot of question on, what is the test? How can it be used? Should we be using it? If we start using it, what will we see? And so that's sort of, where we stand is kind of trying to advise industry what they need to do the testing. What will you see? And, what can you expect? Because if they start using it, then they want to have, more positives than just 0157 alone. And they're going to want to be able to determine, what do they have to do with that product that might be implicated.
So, being fast and accurate is very important. So what is needed to increase the speed and accuracy of these tests?
So to increase the speed and accuracy, the tests are, use PCR pretty much just to look for the gene. And you can't really change the speed there. The machine runs and amplifies the DNA as fast as it can. It's usually on the preparation side, collecting the beef sample, processing that beef sample, getting it into some media, getting that E. coli to grow to a detectable level. Those are the things that they're actually working on in the lab now, is coming up with ways, can we culture that piece of beef for 4 hours, 6 hours, 8 hours, and get it into a detection test as fast as possible, and then, so that's where we're working on that. And then, accuracy comes into how reliable is the test? Is it giving us, can we culture confirm that sample is positive? When we look at the screen, I guess, we did a study where we compared, 5 or 6 different STEC detection methods, across 500 samples that came from a service lab. And we found, each test method we used found, 20% of those samples positive, but they only all agreed like on 2 samples. So, and one was the only true culture confirmable sample there. So, that was good. They all found the positive, but they disagreed on so many other samples that, that's where the challenge is in all of this. And so you want, finding accuracy and coming up with some way of being able to say this is positive, diverting that, meat to a cooked stream of some sort so that, it stays out of, out of the raw, out of consumers' hands and, is made safe.
So what are the areas of opportunities when it comes to STEC detection, testing, et cetera, for, the near future and for farther out?
I think there's we looked at pathogenic E. coli tests. There's a few other markers or virulence factors. I think a number of companies are trying to follow that model now that it's been put out by bioMérieux, who made the PEC test. And so I think we're going to see more virulence factor focused testing, giving us more reliable detection of those STEC. In the short term, had conference calls and things with companies looking for what targets or what things can we use to improve testing for STEC. And then further out in the future, I could almost see us, instead of targeting these genes, we may just take a sample of beef, enrich it for some amount of time, and just sequence everything that's present. If we can get sequencing condensed down to a very, even more economical approach than it is now, we can, find and assemble pathogens if they're present, their genomes there. And so I think, that's sort of, the distant future, but, currently I think we're going to see more and more of these virulence focused tests that can give us better accuracy for identifying these E. coli.
Well, to wrap us up, is there anything you want to touch on that we didn't discuss?
I guess maybe on the, we've been talking so much about detection, and I've mentioned culture confirmation side, and that's the other sort of growing area of improvement is, 015787, I mentioned, easy to culture. It doesn't ferment sorbitol. So if you put it on a plate of agar with sorbitol, the colonies don't eat that sugar. They're easy to see. The other STECs, there's a few media out there that help identify them if they're present. FSIS uses an agar called Rainbow, and you see purple and pink colonies of E. coli, and you pick those and test to see if they're the one that you're after. There's a chrome agar. This is another media. Your STECs can be pink on it, and it helps, is very selective, because when you try to culture these things, you get lots of stuff growing there, and you want just a single colony that's your E. coli. And then there's a new product we've found that's been shared with us. And in this case, we're looking for Shiga toxin E. coli. And if those E. coli are growing on the plate expressing Shiga toxin, there's actually a zone around the colonies that make Shiga toxin. And so we're looking at this going, wow, where was this all our lives? Because really lets us, focus in on finding things that are STECs and improving our rate of isolating these things and having more of a reliable confirmation side.
It's time for our famous 3.
Well, to wrap us up here, I've got 3 questions, and the first one is what is your go-to highly recommended beef or food safety resource that you would recommend someone utilizing?
So I guess on the food safety side, there's probably, Food Protection Trends is a great journal that covers lots of variety of things. And then there's also a couple good textbooks out there that I and a number of our folks in Clay Center have contributed chapters to on everything from pre-harvest to post-harvest and each of the pathogens, E. coli and salmonella and control. So those are kind of where I usually, if I need to find facts.
What's a non-ag book that you highly recommend someone reading?
There's actually a pair of books that I really enjoy and kind of go back to them. They're called 1491 and 1493. And they're basically, taking that year that, we say, Columbus discovered America in 1492. And it looks at and describes, the civilizations and the cultures before that point and then after that point. And they're talk about, how many peoples and civilizations were here. A lot of, it does have some agriculture in there because it talks about, the crops and the things they were growing. And then after 1492 starts talking about the trades and how, certain things like sugar and rubber kind of shaped our modern society that we don't really see, so those are probably two of my more favorite books to kind of go back and look at.
So what, in your opinion, what sets apart a successful beef or food safety professional?
So I think part of it's probably communication, which I don't know if I excel at very well, but you need, when you're if you're looking, say, in the beef industry, the most successful folks there are the ones who can talk safety to management all the way down to the guys working on the line where this is important. We found this, making certain that the folks who are calling the shots and doing the budgeting and things and making decisions for how things are going to run, how they understand it, but then also being able to communicate its importance and explain to the guys on the line on how and what needs to be done. So I think having that skill set to be able to communicate across the board is probably one of the more important things there.
Yeah, it's important for almost all aspects of what we do. And so especially the ability to translate science, high-level science, down to common, easy to pick up and utilize in, whether it's beef processing, plants, etc.
Yeah, it's a bit of a challenge to just kind of share it in this format without having, my slide deck and a pointer.
Yeah, great. Well, Mick, thank you for the time and your expertise and some of these issues with STEC and pathogens and some of the challenges we're faced in the beef industry.
You're welcome. Thank you for having me.